Hello, everyone, and welcome to Lunch Break Science. I'm Ariel Johnson from the Leakey Foundation, a nonprofit dedicated to supporting human evolution research and sharing discoveries in programs like this. We'd like to thank Anna Gordon Getty and Kimberly and George Smith, whose generous support has made this episode of Lunch Break Science possible. Here with us today is Leakey Foundation grant recipient and Science Executive Committee member, Dr. Ann Stone. Ann, thank you so much for joining us.
It's a pleasure. Thank you for inviting me.
Ann is a Regents Professor at the School of Human Evolution and Social Change at Arizona State University, and today she'll be discussing pathogens and human evolution. For those of you watching us live on YouTube, Facebook, and Twitter, you can leave questions and comments for Ann at any time during the episode, and she'll answer as many of them as we have time for at the end. Ann, how do we know what diseases our ancient ancestors had, let alone that they even had diseases?
Great question. So what types of diseases affected our ancestors and how do we know? First, what we can do is try to infer which pathogens affected us from looking at the fossil record. However, we don't really see much evidence of disease. What we see is arthritis, worn teeth, abscesses, periodontal disease, missing teeth, which you can see here on the individual from La Chapelle-aux-Saints. We also can see trauma. But, most diseases don't really leave a mark on the skeleton. Very few do. So we can also make inferences about our past evolutionary history from modern and historic hunter-gatherers, and also from our closest relatives, the other great apes. And so if you think about what kind of pathogens might have affected them in the absence of medical care, we think about parasites, so from uncooked or partially cooked food, from water, from the environment. Although, keep in mind that great apes and hunter-gatherers are very mobile across the landscape, so not a lot of waste is piling up, as you see with settled populations. We also might see zoonoses on occasion. So these are diseases that are pathogens that are acquired from another animal. So these would be things like influenza, Ebola, rabies, monkeypox. We could also see vector-borne diseases, so things like malaria. So these are usually insect-borne. So it would be like malaria, dengue, perhaps plague. And then finally, we sometimes see diseases that are chronic. So pathogens that cause long-lasting illness, and sometimes these do make changes in bones, and they may also be latent. So this could include arthritis or something infectious like tuberculosis.
That's so interesting. I know we've seen a lot of bones that have that kind of have the signs of disease in them. Oh. And
sorry.
So you're an anthropological geneticist and use population genetics to answer questions about human and primate evolution. What is population genetics, and how does it help us address questions about human evolution?
Sure. So population genetics is kind of what it sounds like. It's part of genetics that studies populations. So within population diversity and among population diversity. And really, it's part of evolutionary biology. It's part of the toolbox that we can, that I can use in my case to ask questions about human populations and also about the pathogens that affect us.
Excellent. So, now that we have understanding of some of the context of our early ancestors, disease, and genetics, let's dive into your talk about pathogens and human evolution.
Okay, great. Thanks so much. So I'm going to talk briefly about pathogens and human evolution, and then I really look forward to answering questions. So you might think, if I'm interested in human evolutionary history, why am I thinking about pathogens? And when we look at our genomes, we can see that the genes showing the greatest signals of selection are our immune genes. And the alleles of these genes are kind of shifting from generation to generation because they're in a race with pathogens. And so here what you're seeing is something called a Manhattan plot, because it sort of looks like the Manhattan skyline. And the red dots are significant loci that are associated with severe malaria. So you can see these are scattered across the genome. And this would be true if I were showing, association with other diseases. So I already talked about how we make inferences about the diseases that affected us during most of our evolutionary history, but we also need to think about how many people there were. So during most of our evolutionary history until really, really recently, human population size was small, probably less than 100,000 people, and a smaller subset of that is actually actively reproducing. And so how do we know that? Well, we can look at archaeological data, so look at the number of settlements. We can estimate carrying capacity based on the environment and the technology that people have. And we can also use genetic data, right? And our genomes show that as a species, we have very little diversity. And from this, we can estimate an effective population size. So this is the kind of actively reproducing portion. So it's not the same as census size. And that says that we are, through time, our population size was really small, about 10,000. And so this starts to change about 6,000 to 12,000 years ago, really at multiple locations around the planet. And during this time, excuse me, people began the process of domesticating plants and animals. And really, this radically changed not only how we get our food, but also our settlement patterns, and it leads to an increase in population size. Now, when you have a big shift like this, it really affects disease ecology, right? So more people means more susceptible hosts. And then people have a tendency to alter their environment, particularly if they're agriculturalists. And so they dig irrigation ditches, they make structures to store grain, and these attract vectors, right? So you'd have increase in mosquitoes, increase in mice and rats. And then you're also living in close quarters with your animals, and your animals, particularly in the early days, would likely be interacting with wild relatives. And so it kind of provides an opportunity for pathogens to jump from a wild animal to a domesticate to a human. And then with higher population sizes, this means that something that is a crowd disease that needs high numbers of people can be successful. So, there's really a process of going from a zoonotic disease, a zoonotic pathogen, to an endemic human disease. And so here you can see kind of one way we can think about the stages. And there are some pathogens that never make the jump. So here you can see rabies. Occasionally it affects humans, but humans don't infect other humans, typically. And then you have something like SARS-CoV-2, which causes COVID, that's made this shift incredibly quickly. And many epidemiologists think that this basically has become endemic in humans. And then you can see things that are in between, like Ebola, which occasionally jumps from animals, causes a limited or larger outbreak, such as in West Africa about 5 years ago, or a pathogen like that causes dengue, which sort of circulates between humans and animals back and forth sort of constantly. Ebola kind of dies out in humans because we take action to do that. So, one of the ways that I try to study pathogens is to use ancient DNA and also to use genomics. And both of these fields, or both of these are really helping us to understand the evolutionary history of pathogens. So we can learn a lot from the modern genome data from pathogens, but many pathogens have sort of a boom bust life cycle, which means that often modern genomes tell you about the latest boom. And also vaccination has really limited the recent diversity of a lot of pathogens, which is great for us, but makes it difficult to infer the evolutionary history. So here I have two examples. On the left by Dux et al. This is a recent study looking at the measles virus. And measles is a great example of a crowd disease. It requires at least 250,000 to 500,000 people to constantly have new susceptibles. And that number of people probably didn't exist among early agriculturalists. It kind of took a while for the population size to build up. But their analysis of both ancient and a bunch of modern genomes, but also some that are a little older, shows that the jump from cattle, the closest relative is rinderpest, to humans probably occurred about 2,500 years ago, at a time when you really start to see larger urban areas and enough people to maintain it in the population. Now, on the right are the results of a study led by two of my graduate students, and this is work that's in press. And this is on Mycobacterium lepra, which causes leprosy. Now, our work, as well as that of others, shows that the most recent common ancestor is older, closer to 4,000 years ago. But Mycobacterium lepra doesn't really require the huge population sizes to be maintained in the population. It's a pretty slow-growing pathogen. What's interesting is the deepest lineages, branch 0 and branch 5, as you can see on the right, are found in the Pacific, in island Southeast Asia and East Asia. But there's a lot of things we don't know, right? So even today, the pattern of transmission for M. lepra is not well understood. We know that it exists, it affects other species. It's been found in squirrels, monkeys, apes, and armadillos. But those strains are sort of interspersed with the human strains. So we don't have we don't know where it came from in essence. So we don't know its origin. Now, as I said, both of these studies have used a combination of modern genomes, but also genomes obtained from pathology samples. So the measles study obtained genome data from a lung of an individual who died in 1912. And our study obtained formalin-fixed, paraffin-embedded biopsy samples that were sort of 10 to 20 years old. But we also used data from previous studies that had sampled individuals who, in the archaeological record, who had kind of the characteristic signs of leprosy in their bones, because that does leave a mark. So you might be wondering, okay, why don't we know more about these pathogens? And there's a ton of challenges that I want to mention briefly. The first is, with ancient DNA, the quantity and quality of the DNA, or in the case of a virus like measles, the RNA is very low and it's very degraded. So that causes problems. The other thing you have to realize is most bacteria and viruses are not known to science. Secondly, pathogen population genetics and pathogen population dynamics are complex. There are issues of recombination and DNA exchange that make inferences difficult. There's that boom-bust dynamic of epidemics that we see. And then the other issue that's sort of inherent with ancient DNA is just the context, right? So you need to know whether your results are because of contamination in the lab, because of contamination by some other non-pathogenic relative that's in the burial environment, for example. You need to think about pathogen load, right? So what is it sufficient to be able to pull the genome out of a DNA sample? And given the part of the body that you're sampling, is the pathogen even likely to be there? Also, a lot of past epidemics maybe were caused by something that is extinct today. So how would we know if it caused, the individual to die? So those are kind of some real challenges that we have. Now, you might want to know, okay, how do you get that DNA out of there? Well, we work in a clean room environment. We extract DNA from the samples, and then what we have to do is take all that DNA and make what's called a library. And what we do is we add these adapters with known sequence to each end of the DNA fragments. So the DNA is in small pieces. It's only about 30 to 100 base pairs in length. We also put little barcodes so that we can kind of tag all the DNA from one individual. And then we put additional adapters on. And this really lets us, grab a hold of the DNA in ways that are useful. And so we take the DNA, we make it single-stranded, and then we can use these probes that are single-stranded DNA that are designed to anneal to the DNA of interest. So whatever pathogen you're trying to grab. And those probes have a little hook on them. It's biotinylated, and this lets you put magnetic beads in there that can hook on. And then basically you use a magnet to draw it over to the side of the tube, and you remove the rest of the DNA, and then you've enriched your sample so that you can sequence it and analyze it, and hopefully you have enough DNA to put the genome together and compare it to modern and other ancient genomes. So that's kind of the basics there. And so the last thing I want to do is just show you two examples of what we can learn. So first, as I kind of indicated earlier, we can get a sense of timing. In other words, when lineages diverged and when the oldest common ancestor of a set of lineages existed. But we can also get a better sense of the original distribution of the pathogen and its diversity. And here I show some older results of work from my lab on ancient pre-Columbian tuberculosis, which showed a couple of surprising things. And first, I should note that tuberculosis is a pathogen that, if someone lives for a while with the disease, they may develop lesions in particularly their vertebra. And so it's one of the few pathogens we can kind of see in the archaeological record. So we found out two really surprising things from 3 individuals with tuberculosis that were about 1,000 years old. And the first was that the ancient Peruvian individuals had TB that was most closely related to seal TB. So specifically, Southern Hemisphere seal TB. And so this was really surprising and told us that we think TB may have originated somewhere in Africa, and probably at some point it got into South African seals, fur seals, and then spread through contact among other seal species, ultimately to South American seals. And then we know that humans hunted seals, and the pathogen likely jumped to humans during the process of butchering or through consumption of undercooked meat. The other surprising thing we learned is that people had thought that TB was very old, but our analyses allowed us to kind of calibrate the molecular clock and show that the most recent common ancestor of the human strains, as well as the animal strains, which are nested within, is only about 3,000 to 6,000 years. And subsequent research by our group, and which is currently under review and in progress, shows that in the Americas, or in South America, the seals type strains seem to move inland and north, probably through human-to-human transmission, but then are replaced by European strains after colonization. So very surprising result. And then finally, analyses of ancient Yersinia pestis, which caused the Justinian plague and the Black Death, can actually show you the process of adaptation from a zoonotic pathogen to a flea-adapted pathogen that was able to cause major pandemics. And so this is work by Rasmussen et al. And they first showed that plague, Yersinia pestis, was much older than we thought. And more recent work has actually pushed it back to the late Neolithic. But interestingly, this work shows that there's a gain. So it kind of gets a hold of this YMT gene in a plasmid, and this enhances transmission via fleas. And it's acquired sometime between about 900 to 1700 BC. And then we also see other changes that really help the pathogen cause bubonic plague. So there's pneumonic, septicemic, and bubonic plague. And it looks like the earlier versions probably didn't cause bubonic plague, but there's further work that's being done on that. So those are a couple of examples of how we can make inferences about that past history. Let's see.
Anne, thank you so much. That was so interesting. For our live viewing audience, please be sure, if you haven't gotten any questions in for Anne yet, to get those in now. Let's see here. Our first question is, can scientists look at the DNA from someone who died from the 1918 plague to infer something about how COVID-19 spreads, how long it will last, and how it can be cured?
Okay, from that question, I assume you mean the 1918 flu pandemic. So, that's that was our last big pandemic. And I think, I would say two things. First, the 1918 flu virus genome was actually the first ancient pathogen genome sequenced. And what it told us was really the process of its adaptation. So flu strains sort of circulate in birds and also in pigs. And pigs are sort of the intermediary between birds and humans. And so pig farmers can be very important senetal people in a sense. So you can see the process of that particular flu strain changing and then moving from a bird flu to a more pig-human adapted flu. And then, of course, socially, it's a really interesting example, and we see the same thing today. In 1918, 1919, there was a lot of debate about closing down the economy and wearing masks, and we are seeing that now as well. So you can use similar methods to look at SARS-CoV-2, which is the virus that causes COVID. And there's a really wonderful website called nextstrain.org that has put together all the genomes, has pulled together all the data, and you can actually kind of watch the phylogeny and the movement. They have some really nice animations that show that. And so very quickly, people started looking at the virus, comparing it to its relatives to figure out, what makes us different, what makes us successful, and what might be, a way to design a vaccine to see whether existing antivirals that we have might or might not work.
We have another question. Let's see, here we go. Do you think there will be any evidence in the archaeological record of COVID-19?
Oh, that's a great question. So
I was wondering that too.
Yeah, so there might actually. So a great example of we know about the Black Death and the Justinian plague largely because people wrote about it, right? But there are places where, so if you look at the Justinian plague, most of the people writing about it are in the Mediterranean region, right? So this is happening, the Roman Empire is kind of disintegrating, but places like England, there's a few monasteries, but most of the population is not literate. So most of what we know from that period is actually through the archaeological record. And what you see occasionally is a mass grave. We don't see that many of them, right? So suddenly a mass grave, it means that suddenly people are not buried in the customary way.
Right.
So when you see a sudden switch in burial practice, that can indicate a pandemic. And after the pandemic, when we think the Justinian plague swept through, we see this sudden change in how people built their houses and where they lived. So they went from more dispersed settlements to being a little more concentrated, and they changed their house style. So if you look at what people are doing now, I don't think we're changing our house styles that much, but there are some mass graves.
We have another question. Alice Corning asks, how are our immune systems evolving to deal with new challenges?
Okay, great question. So, the immune system, well, it sort of changes as we age, right? We're exposed to pathogens, and there's a lot of pathogens that traditionally, if you had them as a child, it would be perhaps not as bad, which is something we see actually with COVID. That's not to say there aren't children that have severe cases. But, with a lot of these, for example, measles and chickenpox, once you've had them, you're immune through the rest of your life. We don't know yet if that's true for COVID.
Right.
But so that's one way that, young people with their young immune systems, the immune system is super complex. I don't want to, dive into that here at the moment, but, so that's sort of building up, sort of the traditional herd immunity within a population. But there are some people that unfortunately do die as the result of a pathogen. And what that means is their genes aren't being passed down to the next generation, right? If they die before they have children. And so what you might expect is that in survivors of a big pandemic, they will tend to have HLA alleles or other immune alleles that give them an advantage to that kind of pathogen. Now, will it help with the next pathogen? We don't know. But so there are studies that are trying to see, how did alleles and immune genes change pre and post Black Death, for example.
Yeah. So we have another question. Let's see here. It is, do you have any thoughts about how SARS-CoV-2 is likely to evolve?
Great question. So it has one or two, I think it has a mutation about once every 2 weeks. But it's fairly constrained. So a lot of changes we see, if you look at the genome, and you can actually do this on nextstrain.org, but they have the genome and where we see mutations. And most of the genome, we don't see mutations. There's a few spots where we do see variants that have built up in the last, 6 months. And the question is, how do those affect the pathogen, right? Do they help it? Now, at the moment, I don't think SARS-CoV-2 needs much help. It's doing really well. So we don't seem to see a difference in transmission patterns among the different strains, but that's something that I think people will be watching. I think the other reason why it's important to watch this is to see whether or not it changes in ways that might help it evade a vaccine. So just to compare two viruses, the measles vaccine, it's been the same for years. Right. It works fine, right? Influenza, we got to get a new vaccine every year. And that's because that particular virus mutates very quickly, and it mutates in the spots that help it evade our immune system. And so to tell, a vaccine is basically America's Most Wanted poster or something, right? It's like, here's what you're looking for, immune system, go get it. And so with, influenza virus, it sort of puts on disguises and changes each year. So the wanted poster has to change.
Yep.
So we don't know yet for SARS-CoV-2. It seems to be a little slower, more like measles, but we have to see.
Yeah. We have another question. Let's see here. And you mentioned in your talk that TB may have originated from Africa. So my question is, where and the dates of the evidence? I'm interested. Thanks.
Okay. So the only reason I say Africa is that most of our the greatest diversity that we see today in tuberculosis strains is actually in Africa. Does that mean that's where it was most diverse in the past, or where it originally jumped into humans? We don't know, right? So what's been really great about the ancient DNA data is it lets us, we don't really have a fossil record for pathogens, other than, we can see if it's left a mark and is there, but it doesn't tell us anything about the, exactly what strain was there. And so by using ancient DNA from dated archaeological contexts, we can calibrate the molecular clock that we use to kind of figure out how often a mutation happens. And doing that, our study in Peru let us figure out what the most likely date for the common ancestor of all strains that affect humans is. And since then, additional work has come out, and we're still at about the upper end of our range, sort of in the 5,000 to 6,000 years ago. What's also fascinating is it looks like it jumped into humans, and then we gave it back to animals, because all the domestic animals and even wild animals that have been discovered with TB have strains that are more derived than the human strains.
I think we have one last question. Are there reasons to think that pandemics will become more frequent?
That's really hard to say. I think, there's a lot of concern as human population size increases and encroaches on wild environments and disturbs the ecologies of those environments, that potentially that provides additional opportunity. It's zoonotic jumps seem to happen every 100 or more years, right? Where we end up with a pandemic of some sort. And unfortunately, it's really hard to, preventative measures are often hard to get funding for.
Right.
And we've seen that, unfortunately. And I think we have to sort of retain the memory of the 1918 flu pandemic and this pandemic to show why it's so important that we do those preventative measures. And those include surveillance, it includes rapid response teams to identify new pathogens when they arise and stamp them out quickly. Because with SARS-CoV-2, in February, I heard a talk about it, and it was really obvious that the genie was out of the bottle at that point. And it was probably in December that we lost the opportunity to really snuff that one out.
We have another question. Do you think a COVID-19 vaccine will be more like a measles vaccine or more like a flu vaccine?
So that's the big question, right? If we're lucky, it's more like a measles vaccine, and both in the sense that it causes long-lasting immune response. So we don't have to have a shot every year because our immune system just maintains its surveillance and can respond. We might have to have some boosters. We do for measles, for example. But if it is changing in a way that it evades the vaccine, then new vaccines have to roll out every so often to say, okay, the virus has now disguised itself again. Here's the new picture. And I don't think we know that yet.
Right. Okay, so we have one last question. This will be our real last question. Is there any way to predict what the next pandemic pathogen will be?
No, but it will probably be a respiratory transmitted virus. And that's just because, so part of the reason why SARS-CoV-2 is so successful is that people who, there's a couple things, but people who have become infected are typically asymptomatic for 3 days or so, where they're walking around, they feel fine, but they're transmitting the virus, right? So nobody knows they're sick. And so that's kind of the perfect opportunity to infect a number of other people.
Right.
And then you get sick and you stay home and take precautions and avoid people. But, if a pathogen hits you very quickly and you get really sick, you're not going to go out and, probably affect a bunch of other people. So usually when people are trying to predict what the next pandemic will be, it's usually something that sort of fits the description of what we have now.
Well, Anne, thank you so much for joining us today. It was really great to hear from you, and it was so fascinating and timely.
Oh, thanks. My pleasure.
If you enjoyed Anne's episode today, please check out her lecture, Tracking a Killer, the Origin and Evolution of Tuberculosis. You can find it on the Leakey Foundation's YouTube channel. We will also be sharing the link in the comments right now. The talk delves deeper into the long relationship between humans and tuberculosis. It was a really great talk. We have only 2 more episodes of our pilot season of Lunch Break Science, and I want to know what you all think about the series and what we should do next. So we're going to be sharing a link to a survey in the chat for those of you viewing live, and in the video comments for those of you viewing this on demand. We would really love to hear your feedback and use it as we craft the next season of Lunch Break Science. If you are interested in learning more about the Leakey Foundation and STONE and how you can help support research like Anne's and educational programs like Lunch Break Science, you can visit us at leakeyfoundation.org. Right now, all donations will be doubled by 2 generous donors, meaning your impact will be doubled. Next Thursday, we will meet Leakey Foundation scientist Caroline Schupley, who studies cognition in wild orangutans. Be sure to subscribe to the Leakey Foundation's YouTube channel, follow us on Facebook and Twitter, or sign up for our newsletter to be the first to hear about upcoming episodes and groundbreaking discoveries in human evolution. Still hungry and can't wait until next week? Check out the Leakey Foundation's award-winning podcast, Origin Stories. And Stone was actually featured in one of our podcast episodes, so be sure to check that one out. Origin Stories is available wherever you get your podcasts. Lunch Break Science is made possible by the generous support of Ann and Gordon Getty and Camilla and George Smith. Thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. Until next week, stay hungry for knowledge. Bye.